All right, so the first part of this chapter is understanding some key experiments to show DNA is the heredity material. So how do we know that DNA is actually responsible for the blueprint of life, and why that's important for inheriting traits in future generations and future offsprings. And so there's three really key experiments that showed this. So of course the way science works is that there's a large number of experiments that are performed, but these are just three of the really key ones that showed that. So this was three experiments separated over 25 years. So Griffith in 1927 showed a virulent strains of Diplococcus pneumoniae could be transformed to virulence. And then Avery, McLeod, and McCarty built on those original experiments in 1944 to demonstrate that transforming principle was DNA. So they identified what the molecular nature of that transforming principle was. And then finally in 1952, Hershey and Chase, in completely different types of experiments, also showed that DNA was probably the heredity material. So how did these experiments work? So Griffith, so again, these experiments provided the foundation for Avery, McLeod, and McCarty's research. And what Griffith did is use virulent strains of Diplococcus pneumoniae that killed mice. Griffith also had avirulent strains. So these are strains that were unable to kill mice, but they could be transformed into virulent strains using non-living material from virulent strains. And so he identified what's called a transforming principle, which could transform a strain from a non-virulent strain, an avirulent strain, to one that was now able to kill its host. And so how did this experiment work? So in the first lane, we have a positive control. So what is a positive control? So like any experiment, any good experiments, you need to have positive controls and negative controls. And so the positive control showed that if you took living 3S strains and injected them into mice, then the mice would die. We also have a experiment which showed that there were strains. So these are the same species, but they lack some sort of gene that's either necessary for evading the immune system or for killing the mouse. And so as a result, if you injected these strains into the mouse was able to live. Now we have our negative control. And so what this negative control did is it showed that if you killed the virulent strain, it was no longer able to kill the mouse. And so we just take these same bacteria, we kill them using heat. So if you take them and put them at like 99 degrees Celsius, these bacteria will die. And so we inject those into the mouse and the mouse lives. So why is that important? Because it shows that there's not some sort of toxin, for example, in the bacteria, that if you just inject these cells and release the toxin into the mouse's bloodstream, then the mouse will die. So there's something necessary about the bacteria being alive that is necessary to kill the mouse. And then finally, the key experiment is to take a mixture. And so what happens if you mix two different sets of bacteria that are unable to kill the mouse and you mix them together? So we take the avirulent strain, and these bacteria are alive, and we mix it with the dead 3S strain that normally is virulent, but now it's killed, and so it's unable to kill the mice. And so when you make this mixture together and you inject it into the mouse, now the mouse dies. And further, if you take the tissue and isolate the bacteria that's alive within this mouse, you find that you've recovered 3S cells. And so this is the idea of a transforming factor. So something non-living is able to transform a living bacteria and change it into something that now has a new phenotype that is able to be inherited in the future offspring of these bacteria. And so, now that we know all the details of Chapter 6, we've covered a lot of details of how bacteria genetic recombination and genetic exchange works. This should not be surprising, but at the time it was obviously very surprising. So that was the first key experiment, and now Avery, McLeod, and McCarty built on this work. So they used various approaches to destroy certain components of the cell lysis, and they found that DNase utilized to destroy DNA actually also destroyed the transforming activity. And so this was one of the first key experiments to suggest that the transforming principle was DNA, not protein or another component of the cell. So this was very similar. So they take the 3S cells. So again, these are the cells that can kill mice, and they centrifuge them down. And then if you put them over a Bunsen burner and boil this, these bacteria are going to die. And so they're actually going to lyse their contents into the liquid media. And so what you're going to be left with is this filtrate that is composed of things like DNA. It's going to have things like protein in it. It's going to have RNA. It's going to have carbohydrates. It's going to have lipids. It's going to have sugars. Anything that you would find within a bacteria cell is now going to be released into this soup. It's almost like a soup of material. So what if you take this soup of carbohydrates, lipids, proteins, DNA, RNA, and now you mix it up with the 2R cells? And so just to remember, the 2R cells are avirulent. They can't kill mice. But if you mix them with this filtrate, transformation occurs, and now the mice die. So this is another example of a positive control. So if you mix avirulent strains with the soup, you're able to transform the cells into virulent strains. So this has transforming activity. So they take this soup, and now they had access to a number of enzymes that were able to destroy certain parts of the soup. So if you take a protease is able to destroy protein. And so, oops, I even misspelled protein. That's embarrassing. So if you take this protease and destroy all of the protein within the filtrate, what happens? So now we mix it with the 2R cells, and again, these cells are transformed and they're able to kill the mice. And so the conclusion from this experiment is that the active factor is not protein. So now we continue on and we treat it with ribonuclease. And so the ribonuclease will get rid of the RNA. And so now if we mix this soup with the 2R cells, we transform them into 3S cells. So again, these cells are able to kill the mice. And so our conclusion is that the active factor of the transforming principle is not RNA either. And then the key experiment was to use DNase, so deoxyribonuclease. So this enzyme is able to specifically destroy DNA. And so now what happens if we take this and we destroy DNA from the soup, and now we mix it up with the 2R cells, only 2R cells are able to come out of this soup. So they're not able to kill the mice. And so the conclusion is that the active factor within the transforming factor is DNA. So that is great, but one final, so it's good to confirm any sort of conclusion, and especially not only to repeat the experiment that was done, but even more importantly, it's important to test your conclusion in a completely new model system. And so Hershey and Chase were able to do that, so we're going to move away from pathogenic bacteria and mouse as a host to a different infection system. And so this is an infection system we've already seen. So bacteriophage, T2, this is a virus, and it is able to infect a bacterial host, which is E. coli bacteria. And so this experiment also demonstrated that DNA, not protein, is the genetic material. And so the way they did this is they used radioisotopes. And so these radioisotopes are atoms that have a different number of neutrons. And so because they have a different number of neutrons, there's two key ways to distinguish between them. So we can take the normal phosphorus that we use and we can compare it to the radioisotope, and this radioisotope will be a different weight. So sometimes we use radioisotopes and we use the weight differences in order to analyze an experiment. And then sometimes we also can use radioactivity. So the reason that these things are radioactive is that the addition of another neutron creates instability, and the atom can decay, and this decay is associated with the release of a photon that can be recognized, or even a particle, an alpha particle or a beta particle that can be recognized by a detector. And so they used two different isotopes. So one of these isotopes is associated with DNA, and one of these isotopes is associated with protein. So as we'll see a little bit later, DNA has phosphorus in it. Phosphorus is a key component of DNA. And so this phosphorus atom should be able to specifically label the DNA within the bacteriophage. Sulfur is used specifically by protein. Oh my God, I just did it again. Protein. And so if we use this sulfur radioisotope, we're able to specifically label the protein. And so how did this experiment get done? So this is the T2 phage. So the phage consists of a head and tail fibers, and so it's able to infect an E. coli bacteria. So here we see a picture with dozens of these phages actually infecting a single host bacterium. And again, we see differences. So we see both ghost bacteria, ghost viral particles that no longer have the DNA, and also viral particles that haven't injected their DNA yet. And so what is able to enter the cell and direct phage reproduction? And so they took advantage of the fact that this particular phage, so this is not true of all viruses, but this particular phage is composed of protein, but it does not inject protein into the cell. So herpes viruses, for example, are able to inject protein. This is just not true for this particular phage. So this bacteria has injected some sort of material into the cell, and then this material is able to direct the construction of new mature phages. And so they took these phages that were unlabeled and they added them to E. coli in radioactive medium, either containing 32P or 35S. And so these phages had 35S incorporated into them. These phages had 32P incorporated into them. And so this is just kind of shown by color. So, you can distinguish the color of either the body or of the DNA. These phages become labeled. And so then we label these phages and add them to bacteria. And so after that, we are able to separate, they were able to separate out the host from the phage ghost. So after the phage was able to infect the cell, we can now remove it using some sort of technique, and that technique is able to separate our samples into the phage ghosts or into samples with the host bacteria. And so what they found was that the bacteria were labeled with 32P, but they were not labeled with S35. And if they look at the ghosts, these ghosts were not labeled with P32, but they were labeled with S35. And so what is the interpretation of this experiment? The interpretation of this experiment is that it must be DNA that the virus actually injects into the host. So the particle is actually made up of protein, though, so this is why we see the S35 and when we look in the ghosts. But the injected material, which we now know is DNA, that must be DNA because the P32 is now present within the host cell. So this was the third experiment that was key to really show that DNA was the material responsible for heredity. So this DNA was able to actually somehow was able to overcome the host defenses and then direct the assembly of all of the new phage particles. So that is the extent of part 1. So just make sure you understand all 3 of those experiments in their glory detail.